This is a special episode of the Stem Cell Podcast, ISSCR Day 1, with Dr. Chuck Murry. Hey, everybody, we are Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. As you all know, yesterday launched the start of the 2021 ISSCR annual meeting, which Arun and I are attending virtually from the comfort of our own home, in Arun's case, my own office. Today and every day throughout the meeting, we will be releasing special episodes discussing some of the meeting's highlights and chatting with special guests, including Chuck Murry at the University of Washington, who we'll be hearing from in just a bit. What's more, Arun and I have taken the plunge to release these episodes not only in their traditional podcast format, but also as video episodes, which you can find on the Stem Cell Podcast website or Stem Cell Technologies YouTube channel. You get a glimpse into the inner workings of the operation here. Please forgive me. I mean, this is me. I tidied up. All right. We're going to kick things off in just a minute, but before we get to that,
if standardization is a challenge for your HPSC-derived organoid models, be sure to check out Stem Cell Technologies back-to-back innovation showcases tomorrow at noon Eastern Time to hear more about the reproducible lung and brain region organoids to enable the study of development in disease. So for the full schedule of live sessions, you can visit the Stem Cell virtual booth. And actually, I wanted to start off a little bit out of order here. I wanted to start off by recapping the new ISSCR guidelines. There was an entire session devoted to this, and there's been a lot of press that's actually come out recently, the reworking of the 14-day rule and that sort of thing. The ISSCR has updated their stem cell guidelines dictating how we should, focus on stem cell research, not only looking at human embryo research, but stem cell research across the board. So basically, this is a pretty nifty panel comprised of 45 different members in this task force. Importantly, none of the members in the task force could be engaged in the work that was actually under consideration. And what they did here in the new guidelines were establish categories of research. So category 1 is stem cell research that's actually exempt from review. Category 2 has stem cell research that's permissible after an adequate review. And category 3 is, as you might expect, prohibited research, stuff that we shouldn't do under any circumstance as a community. So in terms of the things that have been updated, the culture of human embryo models has been moved to category 2. In large part, thanks to advances in the science, which we've talked about a lot here on the show, these gastroloids, blastoids, iblastoids, all these things that enable advanced study of human development in a dish. But the important thing here is transferring any of these human models is under category 3. So if you're working on some of these blastoid models, you can keep doing that and, have the appropriate review to enable working on it. But as soon as you think about putting it in vivo systems, that's a no-go. So that makes sense to me. And next up, in vitro generation of gametes. This is something that varies based on the category. Again, if you're talking about introducing in vitro derived gametes into in vivo models, into early embryo models, that's a no-go. So that's a category 3. Organoids in general are category 1, including brain organoids, which I thought was interesting, actually. And perhaps this is something we can talk about with Dr. Madeline Lancaster. We've had a lot of concern and a lot of discussion about whether these brain organoids are actually maturing and developing quote unquote brains in a dish. But I think the committee has concluded that it doesn't matter what sort of organoid work you're doing, including brain organoid work. It's all category 1. So it's all good to go. In vitro chimera work is in general category 1. But again, if you transfer things in vivo, it's category 2/3. Embryo genome editing with CRISPR, TALE, and all of these kind of next-gen technologies. If it's for laboratory purposes, it's category 2. But again, sounding like a broken record here. If you're dealing with clinical research and anything approaching in vivo, it's category 3 and impermissible. I thought the important thing here was the embryo model. So like updating these guidelines based on all of these exciting new technologies that have come out in the field in stem cell research. And honestly, listening to some of these talks and some of these sessions, there was a reflection that perhaps the 14-day rule has been held to an unnecessarily high standard, right? Maybe it's just something that's been around for so long that we've just established it and cemented it into our field and into stem cell biology. But if you reflect on the biology itself and the initiation of the primitive streak and so on, it's really much more ambiguous than that. What I'm trying to say is there's no specific reason as to why the 14-day rule should be viewed as, concrete. And I think that's what these updated guidelines are showing, that there is a lot more ambiguity in the science, especially with all these new technologies coming out. So I think these guidelines were due for an update.
Yeah, it's been some time, and I think with the progress of the science, we're going to need to update these things more regularly just to keep up. But I liked a few things, a few elements that became clear is that, one, there's distinctions within each of these categories, like category 3, A and B. I like that because it's provides some rationale. There's 3A, you can't do it because it's not safe. And then there's 3B, which you can't do it because there's really no rationale, or it's just wrong, yuck test wrong. So I like that there was important thinking, and that's critical, I think, when we explain that to lay people, right? Because they want to know why. They want to know why, what's 14 days? What's the meaning of that day? What's the relevance? And lastly, I also liked that there was this element that it's not so cut and dried. These aren't laws, these are guidelines. And whether or not you can do a thing really comes down to the local authorities, right? There's sovereignty, or scientific sovereignty, you could say, for every, I wouldn't say institute, but in some ways, yes, given the IRBs and the local committees. So I think it's trusting scientists to be scientists. It's also informing the lay people in the community, and also deferring, I think, to the cultural distinctions amongst different communities, scientific communities, as well as just, societal beliefs and ethos. So I really think it was a great thing to set off the meeting, just, as to set up those mileposts for what is, I think, what the consensus is. And I think it was emblematic of maybe, I don't want to call it a society in transition, but, talking now about the presidential plenary with the introduction from current president, outgoing president, Christine Mummery, I think there was a real emphasis on how this meeting represented a lot of change. Obviously the COVID, it's clear, and what that did with the virtual format. And I'm sure we'll be circling back to the virtual format many times. I mean, the gamifying thing, there's a lot of things I love, to be honest. The gamifying thing, I'm currently in 0th place. I know you and I talked, you're in 0th place too, but that hasn't clicked for me yet. But apart from that, I think they really have done a lot to optimize the format, although the connection is a little bit spotty. Again, we'll circle back to that. But then also Sally Temple, who's the program chair, she talked about how they redesigned the format a little bit to kind of spread out the talk, so there wouldn't be so much running around. Even in the virtual format, it was a bit stressful to hit all those concurrents. So it's less concurrence. You can make it to more sessions. The idea being also that you'll kind of wander into subjects that maybe aren't your primary emphasis. There were 5 major themes: tissue stem cells and regeneration, new technologies, modeling development and disease, cellular identity, and clinical applications. And I think also, given the past year of societal upheaval and I guess revision, there was, I think, and there's going to be a heavy attention to societal questions, ethics, morality, given the guidelines, that we just talked about, but also inclusion and equity in science. I think we're gonna come to that, so I'm looking forward to that. Just by way of introducing the actual talks in the plenary, I was very delighted to see that Dr. Mummery invited all the plenary speakers to share the aha moment in their careers or in their scientific story, which I will say with all modesty, I think, we did a little bit to establish the aha moment as a nice bit of narrative amongst the scientists who join us on the show. So I was pleased to see that they borrowed a little bit from the Stem Cell podcast there. And it was just an exciting round of talks. What's in a germ layer was the query or the prompt there for the presenters. I don't know, Arun, what do you think generally of this session?
Yeah, I thought it was a great session, good way to kick things off. I mean, Dr. Mummery, after her introduction, after her welcome, wanted to note that Nancy Whitty is actually going to be retiring as a CEO of the ISSCR after 15 years, and who has been absolutely instrumental in getting this society to be where it is today as the premier society devoted to stem cell research around the world. So there were kind words from all past presidents and a whole who's who list of stem cell biologists, including Doug Melton, George Daly, Christine Mummery, Deepak Srivastava, Irv Weissman, Hans Clevers, who was walking through a forest during his particular thing. So that was interesting. A bunch of esteemed dignitaries in the stem cell field. So I thought it was a really nice thank you moment and a really nice send off for Nancy Whitty. So yeah, for sure, this idea of the eureka moment, this is perhaps partly inspired by the Stem Cell podcast. I don't know, but I think it's an important note for the trainees to pick up on. We were all trainees at some point, and there's some inspiration for all of us as to how we got involved in this field in the first place. And I thought it was cool, actually, that Christine Mummery mentioned that her eureka moment, her aha moment, is the same as mine, actually, beating cardiomyocytes in a dish. So I got one thing in common with Christine Mummery. That's cool to see. In general, I thought it was a great session. Robin Lovell Badge received the ISSCR Public Service Award in part because of his involvement on updating the ISSCR International Guidelines on stem cell research. He also actually worked on the WHO Committee for Human Genome Editing. And then diving into the talks themselves, Chuck Murry presented some unpublished data, building on his decades-long mission to restore heart function by introducing stem cell-derived cardiomyocytes into in vivo and ultimately into human hearts, which is something that's in clinical trials. But he's had a lot of focus on introducing these cells into non-human primate hearts. And unfortunately, as we'll discuss, the issue has been arrhythmias, generating heart rhythm problems and issues after introducing these cells into non-human primate hearts. But he introduced some unpublished data from Project Medusa, pretty nifty acronym there, where they were able to basically knock out a bunch of different ion channel genes critical to cardiac electrophysiology in a custom stem cell-derived cardiomyocyte line. And by introducing this line into non-human primate hearts, they were able to eliminate some of those arrhythmia issues. And I think the ultimate concern next, or the next step, is see if these cells are actually functionally equivalent to wild type. So I think Chuck Murry has a lot of work to be done in that particular area. Next up, Mary Hutch, who focused on adult tissue-derived organoids for non-intestinal tissues, Lorenz Studer, which I thought it was a really cool talk because he wasn't talking as much about his dopaminergic neuron work, which everybody knows about from his work with Blue Rock and so on. He was actually talking about the enteric nervous system and ways to actually derive enteric neurons from the sacral and vagal neural crest, and presenting a lot of really cool unpublished data there. And then finally, wrapping up with Dr. Elaine Fuchs, who unfortunately had some technical issues, as we all have in the last year. It's not just her. We've all had Zoom-itis and Zoom fatigue and Zoom glitches that have happened. But she was able to recover and present a great talk on understanding the memory of epidermal stem cells and all things related to the skin.
Yeah, I mean, here we are a year later, and we can't get through the first day without a total debacle. But she did recover like a champion. And for me, the highlight was, Lorenz, as you said, his talk had like 3-4 different stories or little kernels that were all beautiful, unpublished. But my favorite part, and you gotta rebroadcast if you haven't, or get it on demand, because you will hear Lorenz in that beautiful voice of his say, and the mice again poop, which is an exciting development. So tune in for that, folks. If nothing else, it's fantastic. I just want to cut to the next day. We can circle back on some of the trainee portions perhaps, but jumping into day 2, in the early session, there was a focus on this is the first concurrence, and I must admit that I was really focused more on the new tech, which was a lot of imaging there, but I did show up for Cedric Blampont's story because he's, he went to Rockefeller, was there when I was there. I always follow him. He's a god in skin, Elaine will tell you. And he had a really nice story, surveying all his work over the last years. My only complaint there being that it wasn't much new data there. I know my favorite thing about this ISSCR is the emphasis on new data, but I get it. I mean, there's a lot of hawks out there trying to photograph and scoop you out. So I get it, said, doing what you got to do. Also, though, some nice stories from a young trainee, in particular out of Tim Schroeder's lab, Nuraz Ahmed. I think I got the name wrong, my bad, man, but great visual story. I love these imaging stories, looking at GATA1, GATA2, downregulation of GATA1. Bottom line there, that it was cell cycle shortening that led to the attrition of GATA2, not the direct influence of GATA1. I love a descriptive story that gives you an insight, so that was cool for me. Then also I caught this talk by Krishnan Padmanabhan. Again, I busted that name. Arun, you should be reading these names for me, my friend. He's in vivo imaging. They were doing like neural cells in the brain with 2 photon marrying human cells with a mouse model on these chimeras. Amazing idea. I think that we got a long way to go there, though, in getting these chimeras and imaging, live imaging. I mean, it's a high bar there, but a respectable effort. And finally, for me, I showed up to the whole session, the concurrence for Scott Fraser, because he's doing some really exciting, tremendous stuff with imaging. And I love a good micrograph. It was like multi-spectral, multi was a talk title. It was an extravaganza of imaging. And the bottom line there was how I think you're combining the optics and the physics with this kind of computer learning. It had this thing called a hyperspectral phaser approach, where you could image just based on almost autofluorescence. He was talking about imaging metabolism in human tissue primary, no genetic engineering, human tissue imaging to look at processes just using autofluorescence, stacking like 7 different fluorophores and labels. So that was really visually tremendous. So for me, the concurrents were rich. I just made it to those were the 4 sessions that I'd like to highlight. And then I got to the second plenary with shucks, what's her name? Sorry.
Cuba de Souza Lopez.
Yes, de Souza Lopez, my dear friend, who I forgot her name. That's how close we are. But I've really long admired her because she's in the repro space, and I love how she'll give a talk and be like, I'm interested in all of it, talking about the just germ cycle, like all of it. She means it. And she is going after primordial germ cells and epigenetics, the whole kit and caboodle. I'm more interested in like follicle development. So I wish I had seen more of that from her, but she was courageous in sharing a lot of unpublished data. And then that led us to Hans, right? Was he in a forest this time, Arun, or did he? I mean, where was
he stepped out of the forest and into his office, but maybe he was back in the forest after his talk. But yeah, Hans Klebers, being Hans Klebers, talked about 2 cool stories that I think part of it's unpublished, part of it is published. First one is bacteria-induced mutagenesis, and how certain bacteria like E. coli can actually induce mutagenesis and ultimately carcinogenesis, and modeling this using their universally acclaimed gut organoid model. And the second part of the talk was actually their SARS-CoV-2 work, looking at gut infection using their gut organoid model, and more recently a genetic screen for critical genes that are actually impacting SARS-CoV-2. And the the other two talks in that particular plenary were from Yache Shu, whose work we've actually covered quite a bit recently in the podcast, looking at impact of stress on hair follicles. We're all graying a little bit more over the last year. I know I am. There's been a lot that's happened, but the impact of stress on hair follicles and hair graying. And the last talk in that session was Sean Morrison, focusing on all things bone marrow related, the bone marrow niche. I want to bring it back to the first day, though, because I do want to, just like you, just highlight some of the trainees who are doing some of the great work, the great meat and potatoes of the ISSCR. Like that's, in my opinion, that's where the heart of this conference is the trainees, because the trainees by default are probably going to show a lot of their unpublished work, and it's really important for them to get their shine on major conferences like these, especially since you can't really do it in person, right? Everything's got to be virtual, so you got to let the trainees shine. So that's what I'm going to do right now. I'm going to highlight some trainees. So speaking of SARS-CoV-2, there was a session in the tools for basic and applied stem cell bio subsection, where there are 3 back-to-back talks from trainees, I believe. Agnesia Ryback-Wolf, who is modeling viral infection, SARS-CoV-2 infection with brain organoids. Tim Blenenskoff, using SARS-CoV-2 infection and in the eye and looking at some of the retinal infection there. And also Jesse Huang from the Cotton Lab that we've actually covered quite a bit on the show, using their iPSC-derived alveolar cells as a model for SARS-CoV-2 infection. And I think, as somebody who's done some SARS-CoV-2 work over the past year, I think it's important to show that there has been an incredible utility for this different stem cell models in helping to study SARS-CoV-2 infection. Not only Hans Klevers with the gut organoid models, but the eye, the brain. Shubing Chen over from where you are, right? You two had a bunch of different organoid models for studying SARS-CoV-2 infection. Jared Churko, who's a mentor of mine from Joe Wu's lab, who's now in Arizona, looking at SARS-CoV-2 infection of cardiomyocytes. So that definitely is a focus of this particular ISSCR, is recapping the amazing work that's happened over the last year, focusing on coronavirus. But in addition to that, in addition to the COVID work, there were some other cool talks in that tools for basic and applied stem cell bio session. Ilya Singek, whose paper we actually covered recently, developed a next-gen ROCK inhibitor. I'm sure you remember that ROCK inhibitor-like cocktail that's able to improve the survival of stem cell-derived products for an extended period of time. You talked about that for a bit. Eric Willems, Technologies Facilitating Genome Editing. Robert Zweigert, as a heart guy, I liked him when he talked about his heart-forming organoids for modeling congenital heart disease. It's just a variety of different amazing talks from the trainees out there, and it's not gonna end today. We've got another 5 days of this stuff, so I just gotta emphasize that. Pay attention to the trainees and go to their poster sessions too. They're not able to be in person, but this is how trainees get jobs and make connections, a lot of times through these talks and through these poster sessions. So let the trainees get their shine, Daylon.
Man, I'm not trying to hold them back. These trainees, they are the meat and potatoes. You said it. They make it go. And at this point, these PIs don't even know the tech anymore. Let's be honest. So if it weren't for the trainees, they'd all be on an island. This has been nice. The very first roundup session from our video episodes. We're about to get into a nice chat with Dr. Chuck Murry, one of the original gangsters, progenitors of cardiomyocyte and IPS research, ES, everything. So that's going to be a fun talk. Let's get right to it. All right, people, we are here with Dr. Chuck Murry, but before we get to the chat, I have a brief message from STEMCELL Technologies. Whether you're attending ISSCR virtually or just want to see what's new, come visit STEMCELL's virtual conference exhibition to keep current with the latest advances and innovations in pluripotent stem cell maintenance, differentiation, and cell therapy. Visit www.stemcell.com/ISSCR to view our upcoming talks and specially curated resources for stem cell biologists. Now, onto our conversation with Dr. Chuck Murry, who is professor at the Institute of Stem Cell and Regenerative Medicine at the University of Washington. Also has a senior VP position at Sana. We're going to get to that. His research focuses on stem cell biology. He really needs no introduction. He's one of the first to get human embryonic stem cell derived cardiomyocytes into the heart in vivo. The lab there has an emphasis on understanding differentiation of the human cardiovascular system and using these cells to study diseases and to regenerate damaged tissues. Thanks for joining us, Chuck. What are you thinking of the conference so far?
The first couple of days have been excellent. A lot of fun.
Yeah, definitely. So why don't we dive a little bit into the work that you actually presented, during your session on the first day. You've of course,
sure.
you've been in the iPS cardiomyocyte, stem cell cardiomyocyte field for a long time now. And Dale and I are both cardiovascular biologists. I actually did my grad school in Joe Wu's lab. So I know all about the stem cell cardiomyocyte field. And of course, there's the big goal is we want to get these things potentially into patients to help restore cardiac function after myocardial infarction. And your lab has done a ton of work on this, in introducing these stem cell cardiomyocytes into non-human primate models. And I think what you showed was really exciting because it was an example of how you can avoid that problem of arrhythmias, right? So arrhythmias are the big issue with introducing these cells into non-human primate models. And you were able to generate these Medusa cells, this cool acronym, I must say, these Medusa cells where you modified electrophysiological DNA to understand and suppress arrhythmias, right? And these Medusa cells, after you introduced them into the non-human primates, were able to basically not induce arrhythmias. Now, the question I have is actually something that came up in the Q&A session. When it comes to actually the function, the overall function of these cells, are they still functional to the same extent that a wild type human cardiomyocyte would be after introducing it into these non-human primate models? So talk us a little bit about that.
Yeah, excuse me. So what we did was basically generate a line of stem cells that would beget cardiomyocytes that we think have lost their pacemaking ability. So they don't spontaneously generate action potentials or beat in culture. But the key thing is that they will follow externally administered electrical stimuli. And so when we put a patch clamp electrode on them, we can get them to go up to 3 hertz, for example, or when we do it field stimulation, we can get them to beat very nicely. And we can watch them beat. So they seem to have normal appearing contractile mechanisms. This is just to the eye. We've not really gone in a sophisticated way into force generation on a per cell basis, that sort of stuff yet. Those are experiments still to be done. But they, as you said, when we transplant them in, they do not cause engraftment arrhythmia. So that was really the gist of the talk from yesterday. And the key question is, will they still have beneficial effects on cardiac function after myocardial infarction? And that's the wonderful thing about the human mind compared to the human hands, that the mind can instantly absorb the information and project another 18 months worth of work before the humans can actually get around to doing it. So we'd love to know the answer to that. I can tell you what I think. I think these cells are going to work just fine. I think we have screwed up their automaticity. I don't think we've done anything that's downstream of the action potential. There's one gene we're a little concerned about, the sodium-calcium exchanger, because it's the main way that cells offload their calcium that comes in during the action potential. So we'll have to see. So job security, we've got some work to do in front of us, but we're optimistic that these cells are going to represent a breakthrough en route to clinical translation.
Yeah, you said it. It's going to take some time to get that answer, right? And it's taken some time. It's been over 20 years.
Don't remind me.
It's been some time. I won't say that it's been over 2 decades. It's been over 20 years. Does that sound any better? Probably not. But it's been a while, right? We've had the human embryonic stem cells and their correlates, the induced pluripotent stem cells in hand for a while. You've been making cardiomyocytes. You've been putting them into hearts for a long time. And listening to your talk, I thought it was great to have it in the intro session because it really had me reflecting on the arc progress over those 2 decades. And, you know, there's been a lot of stumbling blocks, clearly. And I think it's taken maybe longer than some people would have expected or hoped. And you alluded to it really early in your talk in terms of the history of kind of cardiovascular stem cell therapy with the, you know, the anniversary studies with the paracrine effect and the bone marrow derived. You know, clearly that was a blind alley, right? And that was a lot of resources that were dedicated perhaps in the wrong direction. But we learned a lot from the negative, maybe more than we could have learned a lot quicker. But it had me thinking about in those 20 years, when you reflect, when we finally have engineered tissue and it's saved a life and extended a life, and it's derived from pluripotent stem cells, and you reflect back on the stumbling blocks over the arc of this field that you invented or played a large part in inventing, what do you think are the major stumbling blocks that have kind of impeded the progress, and that the reasons why we haven't really gotten closer to the finish line to date?
Oh, that's a great question. I mean, the first thing was we didn't know how to differentiate the cells, right? Everybody would show a little corner of a embryoid body that had beating cardiomyocytes in it, but when we got around to quantifying it, we were getting under 1% cardiomyocytes. So we really had to stop making teratomas in the plate and really learn how to apply the rules of developmental biology to get reasonable cardiomyocyte cultures that could be turned into cellular therapies. So that was the first thing. And then when we got around to transplanting these cells, most of them died. And so we got essentially no engraftment to start out. And it turns out the infarcted heart is a stressful place to put new cardiomyocytes in. And so we had to learn the rules of what was causing cell death. And then we created what we call a pro-survival cocktail to keep the cells alive after transplantation. So that seemed to be good. And we then started making fairly rapid progress going up the food chain from mice to rats to guinea pigs. And in guinea pigs, we were interested in the electrical properties. And we found that it actually suppressed arrhythmias in the guinea pig, didn't create arrhythmias. And so then we made the leap 9 years ago now into non-human primates. And it was wonderful because we showed it's possible to do large-scale remuscularization of a heart in an animal that should be predictive of a human being. But we came into this problem of engraftment arrhythmias. And so it's been like peeling an onion. You know, there's one thing, and you can only solve, as soon as you solve the first layer, you come to the next layer of problem and to the next layer. And the thing that we know that still is in front of us is the immune system, because we're looking to do allogeneic off-the-shelf therapies. And we need to be finding ways to get around the immune system with the vision of being basically like the universal donor cell, by engineering the cells to be hypoimmune. And that was one of the reasons I brought my work into Sana biotechnology, was because Sonia Schrepfer from UCSF has also moved into Sana, and Sonia's got an outstanding program in stem cell immunobiology. And I wanted to partner with her because I thought that would be the greatest chance of success.
So what about the maturation side of things? Like, I'm an in vitro cardiomyocyte biologist. I don't do very little in vivo work, but this has been an issue plaguing our field for so long, right? The idea of making these cells more and more adult-like, more mature electrophysiologically, functionally, and so on. And of course, over the years, there have been a number of approaches to actually further promote the maturity of these cells through functional pacing and all this kind of stuff, metabolic maturation and all that. But for the in vivo perspective, is this something that is critical? I mean, for the work that I do, perhaps it is really important to replicate an adult myocyte outside of the body. But for in vivo transplantation, is it okay to put an immature cell into the body and assume that it's just going to become mature due to the niche that it's placed in?
Well, that was the assumption that we went with for many years. And let's set arrhythmias aside for just a moment. If you just look at from the cell's perspective, it works pretty well. These cells start out as little round balls, and they gradually spread out, elongate, and turn into adult-like rod-shaped cardiomyocytes. They polarize their junctions to the intercalated discs and that sort of thing. And so after several months in a proper in vivo environment, they really look quite good. And so the most adult-like we've been able to get a stem cell-derived cardiomyocyte has been through in vivo transplantation. Tissue engineering approaches with the kind of electrical simulation that you mentioned are probably next best kinds of things, certainly the best in vitro. So is it reasonable? Yes, but, and the but is that we have this issue of this awkward adolescence stage where the cells are immature compared to the surrounding cardiomyocytes, and they've got these extra currents that they have no business having in that environment, and they're missing at least one current that we think they should have. And so that was the basis for our gene editing approach, was to try to tinker with the genome so that we could induce a more adult-like electrical phenotype in our cells prior to transplantation. If we'd love to know how to do that more organically, more naturally, by taking advantage of natural developmental mechanisms that drive that process, that remodel the ion channel gene expression repertoire. We, like many others, are working on metabolism, we're working on transcriptional regulation, we're working on biophysical kinds of things. And it moves in that direction. You get the vector, but we're not nearly down the road developmentally as far as we need to be yet.
Yes, and to get down that road, I mean, you just alluded to it. You've kind of gone in not exclusive of your academic involvement, but you've joined Sana Therapeutics with Sona Shrepfer there, and you said it's really to get to the finish line, right? It's to try and get the resources, both intellectual, I'm sure financial as well. But a scientist that I really respect told me, in forecast, and he makes some pretty bombastic statements from time to time. So, I mean, take it with a grain of salt, but he said that the future of mechanistic science is in industry. And that's not to say that you won't find mechanistic science in academia as well, but I think the idea that he was trying to get at is that in order to translate, in particular, cell-based therapies, you're going to need to tap industry, but we always knew that, to get any therapeutic from experimental to commercial stage. But not only that, but in industry, it's not just going to be about the endpoints anymore, it's going to be at the mechanism. Do you find that there really is an emphasis on mechanistic science in industry, more so than before, and that's what's really leading a lot of academic scientists to partner with industry? Or is it the same influences you think as historically, resources, financing?
Really, really an interesting question. I think one of the pleasant surprises I've found joining Sana has been the extent to which they're supportive of mechanistic science. This is mechanism with a mission, I guess I'd call it, because there are things we simply need to understand if we're ever going to rationally optimize them. And so this is a whole new kind of medicine. And this is sort of like applied developmental biology, right? And so I think it's very exciting because we have industry level resources and we're tackling fundamental problems of really developmental biology, maturation, injury and repair, things like that. But it needs to be very focused, of course, with an eyes on the prize. Like, this is what we need to know in order to make something work in the clinic. So if you're of the mindset where that kind of, mechanism with like bumper guards on the bowling alley, that sort of thing, so that it tries to keep your vector pointing towards the clinic, it can really be quite an interesting place to work.
All right, Chuck, I wish we had more time to share with you, but we got to let you get back to the conference. Just before we let you go, I just want to ask, anything in particular you're looking forward to? Any talks, any presentations or sessions that you're really psyched about this year?
Oh, I mean, the one that we had this morning was really fun on the stem cell niche. I mean, from Yashay Su from Harvard. I now know why my hair goes gray and falls out when I'm stressed. And so that was an interesting one. The fun thing about this is that you just stuff pops up in all these niches. Let's say where I wouldn't where I don't spend most of my time. And so you just get these really fun, interesting things that, sort of make you, it's good for party conversation and stuff like that. So it's been a fun conference.
Yes, so far we're only through the first day, now midway through the second, but that brings us to the end of our first ISSCR 2021 video episode. Thanks for joining us again, Chuck. Don't forget to follow us, listeners, watchers in this case, on Twitter at STEMCELL Podcast to find out what we're doing at the meeting. And check back here tomorrow for our next episode with Dr. Madeline Lancaster. That's going to be really exciting to talk to her. We'll see you then. You can see us then and hear us too. Thanks for joining us.